basically, I'm just going to give you sort of basic principles here. Okay. I think as we go through the analyses, hopefully it'll become a little more evident things that you build on to get better designs. So the things I'm going to talk about are replicability, objectives, getting from the organism to the data, and then the steps in designing the study. So the basic tenet here is that if it's worth looking at, we can replicate it. And we all heard about cold fusion and what the problems were with those sets of experiments and how they weren't replicable. But the other thing you have to remember is that just because something is replicable doesn't mean that it's worthy of investigating. And that's what happens when we inject artifacts into our experiments. And then we can always get back those artifacts, but it's not biology. Now, the point of the statistical analysis and the statistical viewpoint of doing experiments is that when we do an experiment, we haven't measured everyone, and we haven't measured everything, and we haven't necessarily measured without error. Okay? If we measured everyone and there was no error, we would never have to replicate. We take one measurement for everybody, and that's it. And if everybody's the same, there's no biological variability, then we only have to measure one individual. So our objective is to overcome basically the main problems that we have, which are measurement error and biological variability. So there's some jargon. A sample is the set of observed items, and we need to know how variable they are. And we might need to do technical replication, but we don't actually always need to do technical replication. Okay? And actually, one of the things that just drives me crazy is the huge waste of resources in the high throughput community in doing technical replication on well-validated platforms. If you are using a sequencing platform or a microarray platform that has been well-validated for your organism, why do you need to show that the thing works? It works. I mean, unless you messed up. Okay? So there is one time when I do technical replication, and that's when when you do technical replication, when you average things out, it reduces the technical error. So if I have a really weak instrument, so it's very noisy, then I do technical replication to average out the error. So for instance, in dilution studies, you often have to do this because the dilution itself introduces errors. Okay? The other time is when what I'm measuring is really expensive, like human brain tissue. Right? You're not going to get a whole lot of tissue from living individuals. So you got to make the best use you can out of the ones you have. So if you got enough tissue in a biopsy, you might want to make technical replicates to get rid of all the noise you can. Even though the important noise is the biological person-to-person noise, you still want to get rid of as much technical noise as you can. But for typical studies where the samples are not that expensive, it's actually running them through the sequencer or doing the microarray, that's the expensive part, I would never do technical replication unless I'm validating the instrument. Okay, so when you're designing the experiment, you need to decide what you want to make inference about, and then you want to make sure that the sample is representative in some sense, and usually randomization is the way to go. So we need to take a sample of several individuals to make sure we didn't you know, the smaller the sample, the more variable, but also the more chance that we're going to introduce biases because we got everybody who was high or things like that. We need to estimate the variability, especially the biological variability, and sampling has to be done at random. If we're trying to make inference about the technology, then what we want to know is, if I measure this something twice, will I get the same answer, right? Or is this instrument giving me the same answer as that instrument? Then we don't want to have biological variability because we're only interested in the technical variability. So if we're making inferences about the technology, we only want technical replication. So you pool huge samples together so you have enough material, and then you just technically take samples from that. But if you're doing biological inference, you definitely need to have biological replication. And some of the worst mistakes that I've seen people do is they'll take samples from 10 individuals, pull them into one big RNA sample, and then make 5 microarrays out of those pooled samples. Well, once you pool it, you don't know what individual it came from, so you don't know how variable the individuals are. If you have 5 microarrays for your 10 samples, pool them into sets of 2 at a time, independent sets of 2, and have 5 pools of 2, so you can get some idea of how variable individuals are, even if you have to have 2 at a time. So when would you have to have 2 at a time? Well, you know, if you have a really small tissue that you're sampling, you might have to pool, although these days the amounts of RNA you can get out and the amounts you need are kind of tiny, so probably you don't have to pool. Okay. So the way I see it is this. The biological samples are at the top. Sometimes there's another level of biological sampling, like if we're looking at normal versus tumor in the same individual. And then everything else is technical. So yeah, we're interested in some of these things, you know, when we're trying to get a good protocol and a good measurement instrument and so on, but doing it twice is just a waste of resources once everything is validated. So you determine the objectives, you figure out what the experimental conditions will be. In statistics, we call them treatments. So if you have a treatment and a control, the control is also a treatment, which is very confusing to medical folks, but that's how we talk about it. You determine the platform, the sampling design, how you're going to do the replication. Nowadays, we have to worry about sequencing depth because typical instruments give us way more sequence than we need for one sample. So we determine what the tissue pools will look like, and then finally, at the very end, we do the study. And then, of course, we do the analysis. Okay, so we need the replication to match the objectives. For differential expression or differential whatever, we're going to look at mean differences, so we need a certain amount of replication. For clustering, actually, the sample size issues are quite similar to differential analysis. And in other cases, we might need to do other kinds of computations to figure out what samples we need and how many of them. Okay. So here's an example of a study in which there's several levels of biological variation we want to deal with. Okay, so we do something to the mice that make them create multiple tumors, and then we might want to ask ourselves several questions. How similar are the tumors in the same mouse? How similar are they in different mice? Are there spatial differences in the tumors? Right? So those are all biological issues. So that's biological variation. On the other hand, if we're not sure how to prepare our samples for our measurement instrument, we might want to look at 2 different sample prep protocols. That's technical replication. So on the de novo transcriptomic projects that I was on, we did a bunch of technical experiments with only technical replication because we were interested in that time. We needed uniform covering of the transcriptome because there was no reference, because it's de novo. And so we needed to make sure there were no biases in the fragmentation and the amplification and steps and so on. So we looked at several different protocols for a model organism, which we could map to make sure that we could pick the best protocol, and that's all technical, right? Okay, so the next thing, hopefully you guys all know everything that's here, but anyway, you have to decide what the treatments will be. So for example, if we're looking at mutant types, we might want to make sure that our wild types come from the same genetic backgrounds. When possible, if we're looking at differences among tissues, we'd like to have we'd like to have a complete set of samples from each individual. Obviously, that's not always possible, but if we can, we'd like to. We need to know what the appropriate controls are. We might need to know what the appropriate dose is if we're adding something, an exposure, right? I just reviewed a proposal where I didn't know the biology. I was supposed to look at the statistics. It looked good to me, but one of the other reviewers said, well, that's fine, except all the animals are going to die because the exposure was too high. Okay, so you got to figure all these things out, and that's why we have preliminary studies, and that's why we review the literature. And then, you know, we have more detail. Like if we're doing a time course, are we going to have the same? Are we going to be able to take multiple observations on the same individual without damaging that individual, and so on, right? And we have to worry about synchronization among different individuals, right? So with yeast, great, we just heat shock them and they all start the cell cycle at the same time. But if you want to look at development of a disease in humans, you got to know when they were infected, and we don't usually know that. So, okay. Some jargon, we usually sort of, a set of treatments that sort of come under the same label is called a factor, like genotype would be a factor, or an exposure would be a factor, and so on. Typically, we only have one factor, that's called a one-way layout, but we might have more than one, like genotype and exposure. That's called a two-way layout, and if all possible combinations are in the experiment, we call that factorial. So a design is said to be balanced if there's equal numbers of replicates at all the levels for all the conditions. Now, again, there's a misconception that designs always have to be balanced. And so, you know, people say, well, the problem is I can only find like 5 individuals who have this condition, and I want to compare them with normals. Well, okay, so get 20 normals, and 5, you know, it doesn't have to be equal. The statistical analysis now can take care of those kind of problems. Okay, so just to make that a little more concrete, this is what the treatments would be the common, in this factorial study, 2 genotypes, 2 tissues, 3 times, that's the set of treatments for a complete factorial. Do you have to do a complete factorial? No. If you know in advance that all these guys are going to die, don't run this one, right? Because you're not going to get any tissue samples anyway. It's just a waste of resources. Or if, you know, if these guys are really expensive to get for some reason, I don't know why. It's a time course, so the first one's usually not the one. It's these ones, right? If they're very expensive to get fewer samples here. That's all. The one thing I should say is the more, okay, if you have statistical support of some sort, like a consulting center or a bioinformatics center, you can do infinitely complicated things and just hope that the statistician will, or bioinformatician will take care of it. If you're doing it yourself in-house, maybe you have a grad student take a few statistics courses, keep it simple, okay? And simple means you try to measure, you try to do complete factorials, you try to keep things as balanced as possible. Okay. Pilot studies, hopefully you guys all know about pilot studies. Somehow, again, people think pilot studies are a waste of resources, but to me, pilot studies are kind of essential, not just for putting in grant proposals, but you train the new folks in your lab, you figure out statistical analysis, you try out the analysis and see if you can get things to work and all that. So it's not a waste of resources. And then you got to figure out the measurement platform. Somebody asked me last night, well, you know, which one should you use, microarrays or sequencing? That's a question, right? You got to figure this out for every lab. I would say every lab probably has to figure it out, but it may be that in some experiments in your lab you need to use the other platform anyway. And people over breakfast were telling me about platforms I'd never heard of and stuff like that. So, you know, it's moving so fast. You know, you got to decide: are you going to be somebody who's going to be on the cutting edge of the technology? In which case, if the new technology fails, you're in trouble. Are you going to play it safe? In which case, by the time the paper comes out, somebody will have used a better technology. I mean, these are really tough decisions for people these days. And so the one thing I've got to say, a lot of people in this room are postdocs or grad students. You got to learn what your lab is losing now, but you got to keep track of what's going on out there because it's moving so fast. People come and ask me questions about stuff I never even knew it existed, you know, and I thought I was up to date 6 months ago. Okay. And as I always tell my own students, if I'm not totally up to date, it's not the end of the world, because 10 years from now, I'll be sitting on a beach in Florida. But 10 years from now, you're going to be at the height of your career, so you darn well better know this stuff, right? Okay. Now the other thing is, you got to do your best to do it right, because especially in the early days, people were it was really expensive, and people tried to cut corners. But the trouble is, you can get it out fast. Of course, you want to get it out fast because it's so competitive. But if you get it wrong, there's a cost. But there's also a cost for getting it right, but too late. Right? So obviously, you're best off if you're on time and right. But the main thing is this one, opportunity costs. So one of the things that we all complain about is them that has gets, right? That is the people who are really already out there with great reputations, they get all the resources. So we want to be that person. But the reason, why does that happen? Well, you know, I'm going to allocate $500,000 to somebody. I want to make sure they're going to get results. And this person I know is going to get results, and that person might be doing stuff that looks really interesting and more innovative, but so far there's nothing to show for it. So anyway, there's opportunity costs, and you've just got to do the best you can. But don't forget that you've got to count some things besides your actual experiment. People do cost money. And one thing lab directors always tell me is, yeah, but I got to pay the postdoc anyway, so I'll just have them do this thing like, okay, so one of the things I used to really love is, even after we had well-developed protocols for microarrays, and most universities would have centers that would do the stuff for them, they didn't want to pay the centers, so they'd have a postdoc do it. Well, I'm sorry, but you know, you guys know that this is not a useful skill for you to have. It's not like it was a training issue. It was just cheap. Well, sorry, it does actually cost you. The postdoc could be doing something more interesting and innovative, right? So there's personnel time, there's reagents, and you've got to take this into account. So we never do the perfect experiment. We always do the experiment we can afford. Okay? We do the best we can with what we can afford, and afford means people, money, time. Okay, so I think everybody knows this, but anyway, what about the platform? Microarrays, especially for model species, they're really very reliable. You really shouldn't have to validate them unless you're really terrible at the bench at doing things. You should send them out for processing to a processing center. It's easy to design them for new species. You do a couple of lanes of sequencing of transcriptome for de novo, and then you send it out to one of the commercial places, and they'll print up a microarray. And I've done this for several species. Even back in about 2005, we got really good arrays. I mean, now they're almost as good as model arrays. They're just limited by what you know about the species. They're really good for comparative analysis, but if it's not on the array, you can't find it. And of course, you'll get differential hybridization due to anything that's different among your samples. Okay, so most of us are going to be using high throughput sequencing for most things. It produces lots of information. It can be applied to anything. You don't have to know what you're looking for. You still have this problem. Actually, I don't know why I put splice variation. Any variation at the transcript level or the genomic level, if you're doing chip or whatever, is going to affect your results. But this is the real thing for most of us. We can't manage the data. I was really surprised when my main collaborator said when they sent out the samples, they sent terabyte hard drives out with it to send the data back, until I tried to download one of the samples from Geo, a sequencing sample from Geo. And after an hour, I thought maybe I should compute how long this is going to take. And there's just, I mean, you just can't up and download these samples. I mean, and then you have to manage them and try to make sure they're clean, and it's tough. So there is still a place for microarrays, I guess is what I'm saying. And they're very cheap now. Okay. So then you have to figure out your replication scheme. Usually there is no need for technical replication, and we'll see that again when I talk about the analysis, because I'm going to go through what's called a linear model, and you'll see how technical replication kind of plays in to the analysis. This is where you get your power for biological experiments. For you people doing human studies, this is ridiculous, right? 3 is like, forget it, you should be doing hundreds. But for those of us out there in the biology world with much more limited funding, you see a lot of stuff out on the web with 2 biological replicates, but that's almost useless. Not quite useless, but almost. Okay. Okay, I think I already said this. Technical replication isn't necessary, but it can be cost effective in some cases, but you still need biological replication. It does not replace biological replication. Sequencing depth. It turns out that the more sequencing depth you have in sequencing studies, the more power you have, and that has to do with the special characteristics of count data. So in count data, the standard deviation is proportional to the square root of the count, but you're looking for differences in counts, so even though that standard deviation is going up, it's going up more slowly because it's a square root than the counts, right? So you get a lot more power with more sequencing depth. But the problem is, that's only one sample. You need multiple samples also. So you've got to trade that off, especially now that people use multiplexing in the lanes. So I gave you an indication already how you can estimate what sample size you need. So we will go over that. One thing that I like to emphasize is, if you can't always do this, but if you can, get enough tissue so you can regenerate your sample if you need to. Because even nowadays, things do fail. I just heard that in New York City during Sandy, lots of things failed and people lost entire samples, but you know you're supposed to have backup samples and all that. I tell people, if you can, don't throw anything out until the paper is published. Okay,
Statisticians can help you improve power by improving the analysis, okay? They can also help you by giving you infinitely complicated designs. But in the end, you have to write the paper, you have to present the work, you have to defend it. So if you have a stats collaborator, great, do these endlessly complicated things. But if you're doing it on your own, or you're going to go to the local consulting center and talk to someone for 2 hours, then go back to your lab, make sure they give you something that you understand. As, you know, like in my department, we train graduate students by having a consulting center. And that student's going to graduate, and 2 years later, when your lab says, why did they do it that way, that student's not available. Okay? So if you can grab a collaborator, you're in good shape. I mean, it doesn't have to be a statistician. It has to be somebody who understands statistics, right? Otherwise, keep it to something you can justify yourself. Okay. Determine the pool of genetic material. Again, all these things have come up in the conversations I've had with people already, so you guys are really quite amazing. So one question is standardization of the samples. And so the question, you know, the typical protocol says get equal amounts of mRNA from each of your samples. But if you're looking at some condition in which transcription gets closed down, that's a really bad thing, because you're not going to find that out because you just got the same amount of mRNA, even though on a per cell basis, it's not the same amount. Okay, so you got to think hard about this step and figure out what it is that what needs to be equal, and if there's a way to measure it in these sort of special cases where weird things are going on. Okay, and actually, in the RNA-seq study that I have for you guys to look at, there's a really interesting situation where a gene totally overexpresses in some of the samples. And so you'll see what that does to everybody else in the sample. And then the question is how to deal with that. Okay. Now the next thing is pooling. So sometimes we do need to pool samples. And pooling and splitting is like a crazy thing to do, you know. So you do as little pooling as possible, so you have as independent samples as possible. The great thing with sequencing is you can barcode and you can pool, but you can still identify the samples. And I've never been at the bench, so I don't know how complicated or costly or error-prone that might be. But if it's easy to do and it's cheap and it doesn't introduce errors, that's the way to go, in my opinion. And that's one thing you can't do on microarrays, that I know of, anyway. So what about designing the actual, how you take the measurements? Well, the simplest kind of experiment has one sample per array or one sample per sequencing lane, but that's expensive, as it happens. But the designs are very simple, and we can do very simple statistical analyses, and I've listed them out here, and we'll be looking at these analyses. Yes?
Is there a saturation problem for a high-throughput sequencing? Like you make too much depth of reads that you actually not find things?
Okay, so the question was, do you have saturation effects in sequencing experiments? Well, you can always get more depth, so from that point, no. But here's the issue in sequencing. There's several issues in sequencing experiments, but one is that if you get a lot of depth, then you start seeing, I only know about transcriptomics, okay, so in this slide. You see things that you wish you didn't see. For example, you see pre-spliced RNAs. And this can cause problems in your analysis, okay? The other thing is if some gene really overexpresses, then you don't know whether everything else got depressed or whether, you know, it's just overexpressed in that situation. So if the patient's sick, for example, immune response genes might be highly expressing, and that kind of thing. So there are issues, but I don't think you have the same
saturation issues that you have with microarrays. Oh, the other thing you get if you have really high sequencing depth is, this is transcription is a random process, right? I mean, there's thermodynamics going on, and proteins are attaching and detaching from the DNA, and every so often, even though the gene isn't expressing, the transcriptomic machinery will attach where it shouldn't, and you get a transcript. So at most sequencing depths, you won't see, you might see one, but the probability is really rare. But if you sequence really highly, you can also get artifacts like that. But a more important problem with sequencing is the mapping, because the mapping routines usually exclude repetitive elements. And so if you have very high sequencing depth, that highly expressed genes will look like repetitive elements. So, yes? Yes, that's right. That's right.
So does that mean you just have to sequence enough so that you get a few measurements for your...
Well, as far as I know, that's your only... So the question was, what if you're looking for something that's expressed but at a very low level? So you are only going to find one or two trans reads at typical sequencing depths. Is there anything you can do? And as far as I know, no. All you can do is sequence more, which might mean more samples or more depth per sample.
There are some biological normalization strategies that try to reduce the input of high express genes, and those are built into most of the, for example, ribosomal proteins. Those are built into the kits that you buy. But you can also try to, there are also potential strategies for doing it in a stronger way. And in the case where you're interested in, particularly in structural changes, for example, alternative isoforms. We don't talk about doing it much with RNA-seq because we're usually interested in counts, but you can actually use selection strategies in the same way that people use them for exome sequencing to get at really the rare variants, the rare variants in the very low expressed genes.
Yeah, so I agree with Sean, but I also disagree with Sean. And the reason where my disagreement comes from is that at least with the labs, I mean, you should get a large sample size, but I don't have a large sample size of labs that I work with, okay? And of those labs, I only had a couple that have done experiments, technical experiments, to see the effects of physical normalization by, for example, allowing the samples to hybridize so that the high expressing genes become duplex, become duplexed, and then you get rid of them, right? That kind of thing. That perturbs the sample. And it adds a lot of noise, and then you can't really do quantification. So if you only want presence absence, those things work well, but if you actually want to do a differential expression type study for these low expressing genes, our experience is you got to be, that kind of normalization doesn't work. Well, that's another thing, and people sometimes capture on microRNA, on microarrays, for example, and then pull off the spots they want and detach them, and then I don't know, I haven't seen it, but that's where you want to do some technical replication first to see what the effects are of what you're doing. So. I mean, people are doing very imaginative things with the technology, but the real question is, are you introducing bias? Are you introducing variance? And what are you finding that's real? Okay. So for one-channel data, we usually, like I said, we're using one-way layouts or factorial designs. Analysis can be a little bit more complicated if there's biological and technical reps, but most of the typical software that people use can handle that. Multi-channel data. So multi-channel data for a microarray means a two-channel array. For sequencing data, it usually means bar-coded samples on a single lane. So the main thing there is whether, well, the main things to consider are whether or not the samples that are in the same, on the same chip or the same lane are more similar than ones in different lanes. That's number one. And number two is when a microarray used to be called die bias, in which now I would just call label bias, which basically suggests that you need to keep track of the labels and keep track of what's been done together. Okay? And when you're designing, I think I have this later, so I shouldn't say it now. Okay. So for two-channel microarrays, there's been a lot of attention to design, such as loop design, reference design, die swap design. 4 years ago, I used to go through all this, but most of us are doing sequencing now. So, the same kind, exactly the same designs are applicable if you're only going to put two bar-coded labels, samples per lane. But you know what? We usually do more than that. So again, so for general sequencing data, multi-channel data, loop design is replaced by, actually it should say incomplete block design. A reference design is the treatment versus control, and die swap is labeled balancing. So what does labeled balancing mean? Well, the idea behind die swap was if all the tumors were red and all the normals were green, and if there was a bias in expression due to the kit or the labeling process that you used for your samples or whatever, you wouldn't know that. You would think it was a tumor versus. The same thing happens when you barcode. So what you should do is the same thing. You divide your biological replicates into the appropriate number of groups, and if you have 4 barcodes, then 1/4 of your tumor samples should be barcode 1, 1/4 barcode 2, and so on. Now, people don't usually do that because, when you're sitting at the bench, it's sort of, you got to keep track of things, but this is the best thing to do. Okay. So, okay. So here's, I guess, a simple example. So here's a 2-channel block. So one thing that people used to do was, so one thing that's really easy is if you just have one treatment and one control, you just always do those together, and then whatever effect there is of the lane or the array, it's equal, and as long as you swap the dyes, you're good. You use a paired t-test, right? An idea that people used a lot for microarrays, which hopefully they are no longer using for sequencing, because in the early sequencing experiments they did, is devoting, they used to always devote one channel on the chip to a control. The waste, they used to have 8, there used to be 8 lanes on sequencing chip, but I think it's gone out of practice to use one for control. It's a well-validated platform. But still, sometimes people put the control sample in every lane when they do the multiplexing across the lanes, and that's not really necessary. Okay, it's just kind of a waste of your resources again. And not only that, it means you have the most information about the control, which is not the one usually of most interest, right? It's the treatment that's of most interest. You can do the equivalent of loop designs. People were sort of confounded with the idea, well, this loop design, if you know what that is, it has 2 replicates per treatment. Well, but I have enough money to have 4 replicates per treatment. Well, okay, do 2 loops, It's not a big deal. But if you do something really complicated, again, you really need a statistician on the team. And I mean permanently on the team, not just there to talk about it, because, okay, bioinformatics stuff gets published a lot faster than stats stuff, but it still takes several weeks, and people do come and go, right? So you should plan the statistical analysis at the same time as the experiment. You should keep track of the things that might be correlated because they come from the same individual or were run in the same channel. You should keep track of other things like which things were labeled by the same kit or amplified by the same kit, who did what, which lab did what, all that stuff that is in your lab book but doesn't always make it into the analysis. It turns out that we can use correlation to help us. So, for example, in the paired experiments, it's the correlation that gives us power because there's less variability in the pair. And if you take that into account, you get more power with the same sample size because the S squared is small, much smaller. And we can build it into the analysis. So the final word is, if you did a good job of all this, then you'll be able to defend it. You'll get it as replicable as you can with your budget, and you can analyze. And although this is the last slide, this is not the last word. The last word is this. If you think you need advice on your experimental design or analysis, don't collect the data till you get that advice, okay? And the thing that's really upsetting to every statistician who works with experimentalists in every field is that people don't come in until they realize they don't know what to do with the data that they have. And, you know, if there's biases in it, if you did something so complicated we can't figure out how to analyze it, that's a little bit late. Okay? So get your help early if you're, I don't know how things are outside the US, but most US universities have statistical consulting centers of some type, and if you don't know how to find them, send an email off to the, if you're in like a medical facility, to Biostat or the public health, and if you're in a science type university, to statistics or mathematics, and find out where those people are.